pathways. Okay. So the last time we left off, we were talking about DNA. And so this is going to be the base sequences in the nucleic acids to be stored as DNA in the cell's nucleus. This will hold your code for your cellular protein production. And that protein production actually then prompts the production of almost everything else in your body. And so it also holds the code for RNA production, by the way. So the RNA production gives you the protein production, actually. A few key discoveries beginning in the late 1940s actually led to your structure of DNA. This was not actually a trivial thing to determine the structure of, because you cannot actually see the exact structure of it, at least at the time. So Erwin Chargaff, noted that the amount of adenine, A, is always equal to the amount of thymine, T, in DNA. And so A matched with T, right? And then the amount of guanine, G, always equals the amount of cytosine, C, and so G matches with C. Right? This actually means that the number of purines always equals the number of pyrimidines, because those are purine-pyrimidine sets. Okay, so your secondary structure was actually that complementary base pairing. This DNA secondary structure was then described as a double helix, meaning two helices that are wound together, a bit like a rope or something. Okay. This was actually first proposed in 1953 by James Watson and Francis Crick. This is not actually right, by the way. It's a common misconception that ends up in most textbooks. Watson and Crick did not actually propose the double helix structure. They didn't even provide the evidence to support it. They actually stole that from a lab that was next to them. And the person that proposed that structure is actually, I'm drawing,
Rosalind or Rosalie?
They were the person they sold the data. The person that proposed that structure was Linus Pauling. Yeah. He had the bases swapped out to the wrong side, though. So he had the bases exactly reversed. Okay. And so Rosalind and that group actually is the one that got the data and actually got the bases in the correct orientation, or at least they would have. They didn't get scooped by Watson and Crick. Not that Watson and Crick did not do a large amount of work in the field that was probably Nobel Prize worthy, but the thing they always get credit for is not what they should get credit for. A double helix, again, can be envisioned as a bit like you've all seen those twisted staircases, right? It looks almost exactly one of those old-timey twisted staircases that used to go from one floor to the other and took up very little space, okay? That's an incredibly good analogy for it. And you'll see it looks almost exactly alike. The two strands both have bases in the center, and so you'll have your bases matching up in the center. And those bases, again, are going to always be purine-pyrimidine pairs, so they're always 3 rings in length. Okay? They're going to have the backbones that will run in opposite directions. So, as one strand goes from 5' to the 3' end, the other strand will then go from the 3' to the 5'. So, they're always going to be the exact opposite in the direction they run. Okay? And so each of the rungs is then going to contain one base from each of the strands, which matches up in the middle, so that they can match up in hydrogen bond. And that's what holds that structure together between the two strands. Okay. They're going to, again, contain one purine, one pyrimidine in this ladder orientation. So the ladder rungs are always about the same lengths, which is 3 rings in length. Okay. The AT and GC pairs, these are called complementary base pairs because they do 2 things. It's a pyrimidine set, right? So they're always 3 long. The other thing that's happening here is your hydrogen bond sets actually match up. And so they'll hydrogen bond together in the correct number of hydrogen bonds in the correct spacing and orientation. Okay? So they snap together nicely. All right. DNA in one human cell contains about 3 billion base pairs. This is actually incredibly long. Are we the most advanced organisms on the planet, by the way? in terms of DNA? Who here thinks we have the most DNA? Or the most chromosomes? Or any combination of most advanced DNA? No. Anybody know how many base pairs we have? How many chromosomes we have? 46, exactly. That's where that 23andMe comes from. That you may have heard the name from. 23 times 2, 46. Your average corn has up in the 200s to 800 genomes. So your average corn is far, far, far more advanced genetically than you. And there are organisms that have incredibly impressive genetics and protein productions. And yeah, in fact, probably our greatest strength as humans is our lack. It's that we have very, very poor inputs. Our vision, our smelling, our hearing, all of those are very poor. And so the cognitive load is very low. So that's one of the theories that's been proposed why we're kind of like the smart ones. Because in terms of IQ in certain areas, we're not. Like your average porpoise has a IQ in the 200s by some measures. All right. So again, these are our complementary pairs. And we are going to form hydrogen bonds. The adenine thymine actually forms 2 hydrogen bonds connecting each of those. The guanine and cytosine actually forms 3 hydrogen bonds connecting each of those. Okay, and so they actually pair very differently. So this right here is the DNA. Note that most toy models that you've probably seen are actually very wrong. Okay, there's a very important structural component of this, which is called the major and minor group. Okay, major, minor group. So you notice it looks a lot more like a staircase that's been wound up, which is a good descriptor of it, or like those old-timey stairs, right? Which really are just like a staircase that's been wound up, right? Or a ladder that's been wound up, because really they have almost no step to them, do they? Okay? And so again, you have the minor groove that goes around right here, and you have the major groove, which is this big part that can fit a lot of stuff in. Okay. That's actually incredibly important for the use of DNA. If it just had minor groove everywhere, you couldn't actually fit anything in here to split these apart, read them, nothing. You actually won't be able to use the DNA well. Okay? But if it was all major groove, it hydrolyzed very quickly. So you actually pack and unpack your DNA all the time. Okay, so right here, these are the elements of it. Can everybody see the backbone? That's going to be your deoxyribose connected between with phosphates, right? And so that's your phosphodiester backbone. Okay, this is what the hydrogen bonding looks like, by the way, for the AT right here. And this one right here is the GC. And as you may have noticed, that even if you got, say, a T and a G that would end up in the same sort of spot and try to hydrogen bond, they're not actually going to be able to hydrogen bond well, are they? And so they won't quite come together properly. Okay? And they'll end up giving a structural flaw, actually, that can be found. Now, this is the tertiary structure of your DNA, and that's going to be your chromosomes. So DNA has a helical twist. We can actually twist it further. Has anybody ever used a hose? Like they're trying to use a hose in their lawn or something? Okay. And so has anybody ever like tried to reel it out and all of a sudden it starts going like this and twisting up and tying itself in knots? And then you have to bend it and twist out? That's supercoiling. Okay? Keep in mind that this is like a wire or hose, right? It has its own resistance because it's a double helix, okay? And so as you get a longer and longer double helix, this thing's going to start supercoiling around into larger coils, just like your wire would, right? Okay? And that's actually referred to as supercoiling. When it does that, that's going to give you your chromosomes. And that's essentially DNA that's been packed away. It's very resistant to being hydrolyzed. Your normal DNA actually would hydrolyze over time and actually split up because of the water environment. Okay? And so this is much more resistant. And when you need that DNA, what you're going to do is you would actually unpack it. These are referred to as histones. They're actually what help stabilize that coiling. You can think of them a bit like, do you guys have those little plastic things you wrap your cords around so they don't tie themselves in knots? Or at least seen them? No? Everybody's aware that they exist, right? Okay. Oh, or the hose wraps. You can also use a hose wrap, right? These are like they're little structures that you can wrap around so that your supercoiling doesn't tie itself in knots and it's more stable in a nice ordered supercoiling fashion. Okay. Okay, and so this is going to be your 3 billion base pairs in one human cell. If we were to actually take that DNA and allow it to be just the normal double helix stretched out, that would be 6 feet in length. Would that function well with your cell? A 6 foot long cord stretching out of it? Imagine that, right? We would be incredibly huge mess. Okay. So these have to coil up. They have to stay almost completely coiled up almost all the time, right? You can only uncoil very short sections of it and still fit it inside your cells. Okay? If you even coiled out even a 1,000th of this, it would be way bigger than the cell, right? Yeah. Like even 1,000,000 of this would already be pushing it, and you'd probably be bigger than almost every cell. Okay? Ever measured. So your DNAs can be separated into 46 pieces that are supercoiled around the proteins that we call histones. Those are those little things to help it coil up, right, into the chromosomes. These pieces of DNA are going to pack into what we refer to as chromosomes. Okay. Your RNA is actually going to be the middleman in the process of creating proteins from a gene. And so it always goes from DNA to RNA to protein if it goes to protein. Okay. Not all RNA actually has to go to protein. Some RNA is actually an enzymatic itself and can make structures. Okay. If you go to certain organisms, they have a lot of those structures that we do with proteins in humans, are actually RNA stuff. It's actually kind of an interesting field, in case you're curious. So, again, DNA is very similar to RNA. Big difference is it's going to be deoxy versus with the OH, right? And that's because the DNA is more stable like that, has less reactive without that oxygen on it. The RNA, on the other hand, is going to, again, be a string of nucleotides, just like the DNA, but we're going to have it with the ribose rather than the deoxyribose sugar. Okay? One of the differences is in RNA, we have uracil, and DNA, we have thymine. Okay? So there is a difference in the bases, but only this one. So A is still A, G is still G, C is still C. None of those change. But instead of T in RNA, we have U. Okay? Okay. So in terms of stranding, your RNA is essentially always going to be single strands of nucleic acid. They do have situations where they bend around in little pins and come back on themselves, or pair with proteins and make RNA complexes with those proteins. But they do not make double-stranded structures of nucleic acids like the DNAs do. Okay? At least not to my knowledge. It'd be interesting if somebody finds that. Let me know. RNAs are much smaller. DNAs typically are much, much larger. Okay? So let's talk about some RNA types and where they fit in. You have 3 main types of RNA that are going to be found in your cells, transforming your DNA sequences into protein sequences. I'm going to mostly refer to this process as it's sort of the normal process. So we're going to go from DNA, RNA, to protein. Okay? And that's probably the process you guys had all heard of before. Yeah. Okay, so the three types of RNA you're going to find in your typical cell are going to be messenger RNA, ribosomal RNA, and transfer RNA. Okay? And actually, they do pretty much what the names on the tins say. So, messenger RNA is going to be in the process of transcription. The process of making a protein from a DNA has two key steps. We have to make a copy of that gene that we can send out and use. And then we have to take the protein amino acids and join them together according to those instructions. Okay? And so that gene copy in organisms that have a cell nucleus is going to be made in the cell nucleus. And that's going to be transcription, where you'll have your DNA's double helix. It's going to partially unwind, so you can read the instructions in that little portion. Little portion usually being on the order of like 10,000 base units. But again, that's pretty small for the overall structure, right? Yeah. And again, this is going to make a complementary copy of that DNA from RNA. Okay? And it's going to make it from one of the strands. The copy is actually referred to as mRNA or messenger RNA. The mRNA is going to be a single-stranded piece of RNA. It'll contain the bases that would be complementary to our original DNA strand, so the one that would match up. Now, do remember that this has U, so if we had an A on the DNA in the RNA, it's going to be the U. Okay? Okay. mRNA is going to be Now, this gene copying, this is actually catalyzed with RNA polymerase. So polymerase meaning making a polymer, and then it's making RNA. Okay? Which again is a polymer of the RNA subunits. So this is the overall process of transcription right here. And in this process, what we have is actually a complex of enzymes that forms around the DNA. And that, remember that grooves I was talking about? We have the major groove. That's why that's so important, is it actually has to sit down in that major groove so you can actually interact with it. And what's going to happen is you're going to have this complex where it's going to unzip the DNA as you go along, and it's going to rezip behind. Okay? And during this process, what we're going to do in this open portion is we'll have RNA polymerase. This is then going to make the newly made RNA here by matching your template strand with your new RNA base. Is everybody okay with that so far? And has anybody ever seen like the machinery that makes like chains? Anybody ever watch like how it's made or something similar to that? It actually works a lot like that. So essentially you got your little chain coming off and you have a little machinery that's just going to press them in. So this matches the hydrogen bonding here. These will match as like ATP, GTP, etc. And it'll use that energy from the phosphates to essentially clamp the new one on, making the new bonds. And then the 2-phosphate group. Kind of a cool thing. You ever see the actual machinery running? It looks a lot more mechanical than you probably think. Because it kind of is, right? All right, so this is the direction of transcription, by the way. What we notice here is we're going from our 3' end right here to our 5' end along the DNA. So again, we're opening up from the 5' to the 3' here. And we're making our new RNA from 5' to 3', because we have to always go the reverse direction of our template. Okay. So let's talk a bit more about the ribosomal RNA and our ribosomes. So our mRNA, we're not going to go into a super amount of detail on this because it can get very, very long and detailed. Okay? But suffice to say that this mRNA in humans is not just put out and used. Okay? It actually does a bunch of processes where it's going to exclude parts of it, put them together, and then send them out. Okay? But overall, this is a good enough part of transcription for you to get the gist of it. Now, what's going to happen here is we're going to send that mRNA out, it's going to go to ribosomal complexes, or what are referred to normally as ribosomes. And those ribosomes are going to be an organelle. You guys know what organs are, right? So organs are structured cells in your body that have specific functions, right? In a cell, an organelle is a structure within the cell that has certain functions. Okay? A number of you probably haven't really encountered that, but anybody that's taken the biology stuff has. So, yeah. Now, this is going to be composed of ribosomal RNA. This is rRNA. It will be a complex made with protein as well. So it's going to be RNA and protein making a enzymatic complex. Okay. Now, it's going to be a place where your nucleotide sequence of mRNA is going to be attached around with that complex. That sequence is then going to be read through, and a amino acid series is going to be deposited down, put together, assembled with peptide linkages. Remember those peptide linkages we talked before? Yeah, the amide bonds. Okay. And so then you'll make polypeptide chains. Those polypeptide chains will come out. They will then fold, and that's going to give you your proteins. Okay? And that'll give you your entire protein. Now notice that really this ribosomal complex is only giving you the primary structure here. Okay? Some of that self-assembles, but there are proteins and enzymes that are responsible for assembling this and other cofactors that are responsible. Okay? And we're going to kind of ignore those because it's too much, right? So let's concentrate on getting the main gist of this, right? So again, your ribosome is going to have two RNA protein subunits. These are going to be the small subunit and the large subunit. What's going to happen is this is actually going to assemble around your mRNA. And so your mRNA binds to one. Then the other comes and binds to it. And this gives you your ribosomal complex around your mRNA. And now it's ready to be read. Okay? All right. So before we had transcription, right? If we transcribe something, then we just directly copy it, don't we? Same alphabet, everything else, right? So you transcribe like a YouTube video into English when it was in English in the first place, right? Okay. So the next step is going to be translation. We're going to translate it into a new language, right? So we're going to take base pairs, nucleotides and nucleosides, right? And we're going to transmit that information into protein. Okay? And so this is going to be what's considered the second step, is that protein synthesis. This is referred to as translation. All right? And so in this process, we need one more RNA. This is the third type of RNA we had mentioned, and that's going to be the transfer RNA. So tRNA. And this is going to be responsible for being the little tags that tell your ribosomal complex which one is which amino acid. Okay? It's responsible for pairing those amino acids to codes of those RNA. Okay? Easiest way to think of them is you go into Walmart, and how's the Walmart computer know what you got and checked out? Has little price tags, doesn't it? These are the price tags of amino acids. Okay? This is the little tagging system that tells that ribosome this is the amino acid that corresponds to that RNA right there. Good? Okay, and so these tRNAs are going to be sort of this compact T shape. They're going to have complementary bases that can hydrogen bond to that mRNA, so they'll fit onto three what are called codon codes, three base sequence codes that correspond to each amino acid. Okay? And this tRNA has kind of a compact sort of clover or T shape if you were to flatten it out. It looks a bit like a T or a cross or a clover. Okay? And so tRNA, again, is going to have a triplet 3-base sequence that's called the anticodon and its anticodon loop, which is going to match up. And so what you'll have is you'll have these little tags that will attach to amino acids. On the other side, they code for different mRNA codons, which are 3-letter designations, 3-base designations, which they can then hydrogen bond to. And that's how you actually transmit that signal of the code of your mRNA to amino acid. Okay, so the tRNA is going to have what's referred to as an acceptor stem. This acceptor stem is what actually binds those amino acids. The only way to get an amino acid incorporated into a growing protein chain is actually to have it coded with these tRNAs. That tRNA is then going to end up in the ribosomal complex, match up with that codon, and then that's going to allow the ribosome to attach the amino acid. Okay. It looks very similar to the sort of machinery you'd see in a factory, by the way. In case you're wondering, you'll notice that they often take the products they want to join together and they put little graspers that can hang off the racking, right? And that's how they actually put everything together. Same thing here. Okay. It's surprisingly similar to a human-made factory. Each of the 20 amino acids has one or more tRNAs that are available to bring amino acids to the ribosome. Certain special important amino acids will actually have more than one code that will fit. There is some caveats to this that we're not going to cover because, again, if you want more depth, please come join me in like a biochemistry or something. Or you can join the biology people and their genetics stuff, depending. Okay, and so here's your transfer to RNA and translation again. So the second step in the process, your DNA, that's translation again. I already did that. I don't know why I duplicated it. Okay, and so this right here is actually a tRNA. From this angle, you can't see the T shape as well, but you can kind of see it a little bit if we were to move it this way. Okay, and so what you have here is, again, you have the acceptor stem right here. That's that 3' end right there. This is then going to form hydrogen bonds and various different linkages, mostly hydrogen bonds, among the RNA right here. And this right here is then going to be your anticodon, which is going to match with your mRNA. So that's going to match the base pairs in your mRNA. Okay. All right, so once we accept an amino acid onto this acceptor stem here, what will happen as your mRNA is being fed through, like a little tape or something, it's going to have these base pairs, the codons, match the anticodons. This is then going to feed through. It's actually a 3 set of codons that feeds through. And these essentially get moved in such a way that they get clamped together for the amino acids. Okay? And you essentially take the different amino acids, clamp them together, and form those peptide linkages. This will allow the amino acid chain, which is that peptide chain, to release, flow off as you assemble it down the side here. Okay, so and this will feed through as you go along. Eventually, you'll hit what's called a stop codon, and that will stop this and release it. Okay, actually, usually the stop codon has repeated pairs to make sure it stops, because there is error. Now let's talk a bit about the genetic code here. So the mRNA is going to transcribe from your DNA. This is going to contain a sequence specifying the protein to be made. A given RNA triplet is going to be called a codon, will translate to a specific amino acid. The genetic code wheel actually shows the codons of your mRNA for the 20 amino acids. What you will see here is the first 2 are very important. The third, not so important. Typically, the third one, you can be almost anything and still get the same amino acid. Okay? And that's because in our codon-anticodon pairing, the first 2 pair the best. The third one doesn't pair well. And so there's error there. If you depended on that, you might get the wrong amino acid. And fairly often. Okay. And so there are some important ones. There's 64 codon combinations that are possible with the bases A, G, C, U. If a new amino acid came along, it would actually have to be slotted in here somewhere. The codons that are UGA, UAA, and UAG, these are all stop signals. You'll notice again, the last one is the one that typically varies the most with these. These are usually repeated in most of these processes, most mRNAs, to make sure it stops and doesn't just keep adding stuff on. So, again, this is a review of your protein synthesis. We have transcription, tRNA activation, and translation that we had looked at. And then termination. And so when you terminate, what's going to happen is you hit stop codons. And what will happen then is the protein synthesis will end. This will usually then detach. You'll be left with your protein to fold and then drift off and do whatever it's supposed to do. Okay. The initial amino acid is typically methionine. This is going to be oftentimes removed from the beginning of our polypeptide. This is actually your start and usually helps you attach to whatever is going to happen next, and then get removed to give you your final protein structure. Okay. Your growing polypeptide is actually going to fold into its tertiary structure. This is going to form the disulfide links, the salt bridges, any of the other hydrophobic, hydrophilic interactions that are going to cause it to fold into that shape. It may need other protein complexes to accomplish that, or certain metals to do that, or cofactors like coenzymes. This right here is a summary of this entire process in a figure. So you can see the whole thing sort of summed up. Okay. So right here, we're transcribing right. We make our mRNA. The mRNA gets processed, cut up, reassembled, comes on down here. That mRNA is then going to have a ribosomal complex assembled around it. Then the tRNAs are going to come with their amino acids. Those will be joined together according to these codes on the codons of the mRNA, anticodons of the tRNA, to assemble together into our polypeptide here. There we go. Got the full process now. So let's talk about some possible effects. We can have mutations, which would be a change in our DNA sequence. So you could have no change in the protein sequence is possible. Sometimes a change in your base will have no effect. It'll be the third base in a codon, and that's the same amino acid, so who cares? Okay. So quite a few changes can have just no effect at all. Only about 2.5% of the DNA in your chromosomes actually encodes for proteins. The rest of your DNA is usually what's called non-gene, or a lot of people refer to it as junk DNA, but this is actually kind of wrong. It's not actually true. A lot of it's sort of safety protocol stuff that helps to get the actual DNA that is going to be processed in proteins, to safely get it to be used and to amplify its effects and have it unfold properly and that sort of stuff. But you do have very large chunks of DNA that are essentially you don't actually need. Okay, they don't code for anything. They're just there to mostly protect the other stuff. Okay, and make sure the other stuff actually does what's supposed to. All right. And so that's the non-coding DNA. There's more than one codon that codes for its amino acid. So, for example, like a codon UUU, you could change it to UUC. Won't have any effect. Wouldn't actually matter. Again, that would still give you your same amino acid in the same sequence. Nobody cares. Okay. These are going to be silent mutations, and there's a lot of these silent mutations that have happened already, and we probably have about all the silent mutations possible in humanity that have happened to humanity throughout its history now. We have so many humans, right? So let's talk about a change in your DNA sequence, something that actually has an effect. So if we have a change in our protein sequence that can occur, and it can have no effect on protein function, okay? This would be a minor change in like a loop or something. So it doesn't really matter what the side chain is because it's still going to come back, make our same beta sheets. Protein doesn't have any major change in function. No big deal. This would be an example of something like AUU mutated to GUU. We're going to have a different amino acid, but it still has a nonpolar side chain. In the position, it doesn't really matter. And so we get an isoleucine. It's going to change to valine now, and it just doesn't have any effect on the protein actually doing what it's supposed to do. Okay. So these two amino acids, again, very similar size, very similar polarity. So usually if you get a change between these two, it just doesn't matter. Okay. This is, again, another type of silent mutation. You've mutated, but it didn't have any effect on it. Well, your parents' DNA probably mutated. It didn't have any effect on you. Okay. So this is going to be a change in the protein sequence that can occur and does affect protein function. And so, again, this one's not going to actually be silent in this case. It's going to have an effect. So, for example, if we changed AUU, we mutate to AAU, then the isoleucine would be changed to asparagine. This would be a nonpolar amino acid replaced with a polar amino acid. And so suddenly it could greatly change the effect of your protein because it could change the structure, right? If you required nonpolars to match up because you're going to end up with this nonpolar region, all of a sudden you have a polar part, don't you? Okay? And it can disrupt that. All right. Other mutations can have negative effects on protein synthesis. This could include a codon changing into a stop codon, or inserting or deleting a base. If you insert one base, it's going to change the entire frame, right? So we'll have a frame shift. And so all your codons change, don't they? If we were to delete one base, we would change all of our codons, wouldn't we? We'd code for an entirely different protein. Later shifts, the triplets that read in the mRNA, this would change the identity of all subsequent amino acids if we include one or remove one base. Okay. Stop codons obviously would just give you a fragment of that polypeptide, that protein, instead of giving you the whole thing. Note that these are not always negative mutations. Okay? Mutations does not have any actual connotation of negative or positive. It's just the vast majority of mutations are normally negative. Okay? And are normally, it's much easier to mutate something that doesn't work than it is something that works better, right? Okay. Let's talk about some of the sources of mutations. So, rarely when DNA replicates, we can have errors. These are spontaneous mutations, and this is just a straight up error of your DNA replication. It happens, doesn't happen very often. Okay? Let's think of it this way, right? If 1 in 10,000 times you have an error, that's really good accuracy, right? But if you replicate 4 million base pairs, then you actually are assured errors, aren't you? Yeah. So you have to have incredible accuracy to never get any errors. And unfortunately, that's just, well, fortunately and unfortunately, how evolution would occur. So we do get errors, spontaneous mutations. Environmental agents can produce mutations in DNA. These are going to be mutagens. Many mutagens are actually what we refer to as carcinogens. And that's because what cancer is it's a mutation in a DNA for a cell that then gets replicated that makes that cell immortal. Okay? So your cell has decided to become immortal. It rejects any death commands by you. It takes up all the nutrients it can get to grow then. And it just keeps growing and growing and growing and growing. Okay? It replicates, passes that on to its children, and we can have the types of cancer that just gives you tumor, in which case they don't split off and spread, or we can have the type of cancer gives you what we normally refer to as cancer, and that spreads off everywhere and kills you. Both can kill you though. Did you know that your cells in cancer though are immortal, and if you collect those cells and just keep giving somebody those nutrients, they'll live for forever? You guys know that? No? Yeah? Yep. So if you ever get offered immortality, say no. It's cancer, okay? Cancer. Okay. So viruses can also cause mutations because they can alter our DNA. In fact, that's how many viruses function, is they inject either RNA or DNA that's then going to be read by your body. And so one common chemical mutagen is actually sodium nitrate. This is NaNO2. This is a preservative in processed meats, actually. The presence of amines, sodium nitrate actually forms nitrosamines when assisted in the conversion of cytosine into uracil. And so it actually changes the identity of C's into U's. Okay? Which obviously then changes how it matches up to other things, right? Because the thing that matches up to you is A, isn't it? And so sodium nitrate is still actually used to preserve meat because it kills the bacteria responsible for causing the illness, which is botulism. And we just sort of hope that we have a small enough concentration that's not going to seriously affect the people that eat it. Okay? If mutations occur in somatic cells, so any cell type other than the egg or sperm, it's only going to affect that individual organism. If it happens in an egg or sperm cell, then it can actually be passed on to the progeny, right? Mutations that occur in the germ cells, that's your sperm and egg cells, of course. These can be passed on to future generations. Germ cell mutations will give you genetic diseases. There's more than 6,000 genetic diseases caused by mutations in a single gene that have been identified. Note that this is not just how you get genetic diseases, it's also how you evolve, right? So if that mutation has no particular negative effect, we would call that evolution, right? Okay, and so this is some genetic mutations right here. Right here's cystic fibrosis, by the way. This one's Down syndrome. These right here are the protein or chromosomal defects. So, for example, Down syndrome has a formation of 3 chromosomes instead of a pair of chromosomes. Usually it's on chromosome 21, though you can have it on other chromosomes, which is possible. This would give you heart and eye defects, cognitive and physical issues. Okay. Anyways, here we have a bunch of different ones. There are many, many different genetic diseases, of course. These are the sort of most common, most interesting ones. This is sickle cell anemia, by the way. It gives you a defective hemoglobin from your mutation. It's on chromosome 11. And interestingly enough, you can be partial sickle cell anemia, which actually gives you resistance to certain diseases, which is why that's still around. Anyways, different ones. So viruses, these are very small particles, are going to contain 3 to 200 genes. These can infect any cell type. Here we see some different viruses. This one's the common cold, which is a coronavirus. Flu, influenza virus. And here's some other ones right here. Hepatitis is actually kind of an interesting one. There's actually a form of hepatitis that A through C, you have different forms of hepatitis, and some of them are basically don't really accomplish anything in you. Some of them make you quite sick and actually quite lethal. So it's sort of a some of these diseases are coming back, by the way, in case you're wondering. Yeah. Because our antiviral agents are not working as well. So viruses can make cannot make their own proteins or energy. They're really more machinery than living organisms in and of themselves. They don't have all the parts that are necessary to do any of these processes we've been talking about. So what they do is they have their own nucleic acids. They're going to use your ribosomes and RNA. Not all viruses have DNA. Some actually just have RNA. But for the most part, they're going to inject their genetic material into you. Then you are going to make copies using your ribosomes and treat that DNA or RNA as yours. Okay? That's then going to replicate that virus within that host cell. And that's going to make all their proteins and the new viruses in that. Eventually, it gets to the point where you would burst the host cell, releasing more copies of the virus. Okay. Interestingly enough, some of these viruses actually have gotten so good at doing this that they've actually been able to essentially incorporate themselves in the organism. An interesting side note to this is viruses just want to spread. That's what they're genetically predisposed towards, is the ability to spread. So viruses don't actually want to kill you. Most viruses tend to go towards less lethal over time. Okay? Because if you die too fast, you can't spread. So most viruses just want to spread. That doesn't mean that's a good thing. But it is. So viruses contain nucleic acid, this DNA or RNA. It's going to be in a protein coat. That's called a capsid. And so here would be their genetic material. This is their protein coat right here. And most of them have an envelope and a protein spikes of some sort. So many of these viruses are going to have protective envelopes. This will envelope the capsid to protect it. Your function of your virus is going to be to monopolize the functions of your host cell to orient them towards production of more virus so it can spread more. Viruses infect a cell when the enzyme in the protein coat makes a hole in your host cell. This will allow the viral nucleic acids to enter. It will then mix with the host cell materials and start being replicated by the host cell. Okay. Not all viruses, in fact, many viruses do not have protein spikes, but protein spikes can be very useful to latch onto and penetrate through your cell membranes. Okay, so if the viruses contain DNA, the host cell is going to replicate the viral DNA. If the viral DNA produces viral RNA, this can make proteins for the virus within your mechanisms. The completed virus particles will then be assembled and released from the cell to infect more cells. This actually has a variety of mechanisms that can be deployed. Most of them kind of amount to just building up and bursting the cell open, but not all. Some are very clever designs that can allow them to actually emit virus particles without destroying cells. So there's some pretty crazy stuff that can happen here. We've also modified viruses to deliver DNA or RNA payloads for us. So that's that genetic modification that you may have seen, which the first procedure in history has been performed to treat a genetic disease, by the way. And it's a $6 million procedure. You can get it right now. Okay? It has been approved to be commercially available. This release often occurs by budding. Vaccines are often inactive or weakened forms of your virus to boost immune response by causing the body to produce antibodies. Essentially, what you're doing in a vaccine is all vaccines kind of work in a similar manner, but there's actually different types of vaccines. As you guys probably know from COVID, because that became a huge argument point, is that essentially all vaccines work by giving you parts of the virus to train your immune system to identify. Okay? Now, how they do that is where the differences during COVID really came into effect and people got very angry. For example, one way of doing that is to actually inject you with the mRNA to make those parts. And certain vaccines during COVID caused some very long-term adverse reactions because of that. Okay? As it turns out, yes, that mRNA can travel to your heart and produce the spikes in your heart. Okay? the old style vaccines literally took the virus, kind of smashed it apart, and gave you disabled virus to work on. Okay, that had been heavily damaged. So antibiotics are only effective against bacterial infections. They're actually useless against viral infections. So you may wonder, like, why can't I take penicillin to get rid of my flu? Penicillin would have absolutely no effect whatsoever. Okay, the viruses don't have any of the parts that penicillin would actually target. So what would happen is if you tried to take penicillin, you just kept trying to take it, you'd actually damage yourself. You wouldn't damage the virus at all. Okay. By the way, penicillin and antibiotics in general do actually damage your kidneys and when given in sufficient amounts. Okay, so it's not something you actually should stay on for forever at levels that would actually be effective to combat those bacteria. There are antivirals now, but they're very, very targeted on specific viruses, and typically they involve inserting something that is interfering with proteins the viruses use to attach to your cells or cause certain things. Okay, but there's actually a very effective one for HIV now, which is pretty cool. It took a long time. It takes forever to get good antivirals and actually figure that out. Hopefully that'll speed up with AI, but we'll see. But it does happen after. Okay, so this right here is sort of your viral infection. So number one, your virus is going to attach and invade your cell. It's going to inject the DNA. Typically, this does not happen by actually the entire virus entering. It more merges or spikes through your cell membrane and then injects you with the DNA. Okay. Then your DNA is going to enter. It can actually get into your cell membrane, or sometimes it doesn't actually need to go into the cell membrane. Like if it's RNA, it can just directly go into the ribosomal complexes. It doesn't need to do the cell membrane at all. And so that viral DNA is then going to get replicated by your same machinery. It does a replication of your DNA, because how the hell do you tell the difference? Okay. And so you replicate the DNA as you normally would your DNA. That DNA is then going to be transcribed. You'll get the mRNA. Your ribosomes will then produce the proteins for it, and you'll make new viral particles. This can actually bud, which would be where you have it merge with your cell membrane and actually pinch off, and that actually leaves your cells alive. It does that. Some viruses, though, do not have this mechanism and actually build up and burst the cells. So it depends on the virus there. There's quite a variety. HIV-1 and AIDS. So HIV-1 is the retrovirus that's responsible for AIDS. That's the acquired immune deficiency syndrome. HIV-1 actually infects white blood cells. This is known as T4 lymphocytes. They're part of your human immune system. Now, if you do that enough, you're not going to have your white blood cells that would normally protect you from other infections, and that tends to kill people. Okay? And that's why AIDS and HIV has a tendency to become lethal. Now, depletion of these immune cells is going to reduce a person's ability to fight infections. Today, people with HIV and AIDS can be treated with what is referred to as a cocktail of drugs. A cocktail of drugs is just different drugs that target different parts of this process. They're all given in a certain ratio to prevent HIV from doing this process, to do its life cycle. And so we're going to prevent that process of its life cycle of infecting and then replicating in your protein synthesis and block that. Now, patients can become resistant to certain combinations of these cocktails. And so over a lifespan, you can modify that cocktail to replace it with different drugs that will also prevent that sort of part of it. Okay? And so this can prolong people's life expectancy. These have gotten so good that you can actually have HIV at non-detectable levels now, and people that would have previously had AIDS, they won't even develop AIDS because you'll suppress it down to non-detectable levels. They can't even transmit if they have a proper cocktail that's properly functioning. And so it's gotten actually really impressive. People can live full lifespans, no real effect at all. Okay? It's not really a cure, though. If they stop taking it, they'll develop full blood. Okay? So recombinant DNA, this is going to involve recombining DNA from two different sources. And so this is where that genetic engineering and gene cloning have come from. It's a genome of one organism is going to be altered by typically splicing in DNA from another organism to give it the proteins from that organism. Whether that is the DNA from just other humans because you're lacking a DNA and that's why you have genetic disease, or whether that's DNA from something else to produce something that kills a bacteria or kills a virus, okay, or makes you more resistant. Sign. Humans have actually been crossbreeding plants and animals for centuries, of course. We've been exchanging DNA for desired traits, whether that's just killing all the plants that don't get tall enough and planting the ones that do get tall enough so you get bigger wheat, or any process, really. And centuries is putting it lightly. We've been doing that probably for tens of thousands of years now, since the early cave paintings. Yeah. The recombinant DNA technique was developed in the mid-1970s. This has gotten a bit more advanced than this, and we can do some stuff that I'm not going to go into here. If you're interested in it, again, there's an entire class, actually, genetics, where you can actually do some of this stuff. It'll be cool. And so the advances in the last decades have been really, really impressive, but a little scary too. Honestly, if you want to see the scary part of it, look up Chinese designer babies. Okay, if you want to see the what shouldn't scare you is look up the GMOs and how they're more resistant to drought and actually have allowed us to feed a population 10 times what was predicted we could feed before, and has saved billions of people's lives. So, though certain people do not like GMOs very much. So let's talk about recombining DNA to produce proteins. So one of our first applications of recombining our DNA, not our DNA, but DNA, to incorporate human insulin gene into common bacteria, which is E. coli. This then allows the bacteria to produce the human insulin protein, allowing us to harvest human insulin and give it to people that their insulin production is not quite up to snuff, which would be diabetes, right? Before 1982, we would actually use pig or cow insulin, or it was harvested for use by people with diabetes. However, we had a lot of problems with this because of impurities in our extraction process. It could actually cause rejection in some people, and so then they would just be kind of doomed. Almost all insulin today is expressed human insulin, though this may not actually be true in the near future. They've made a insulin-like structure that can actually modulate its own function in people's blood. So there's kind of a cool thing that might be replacing this in the near future, but for now, this is the insulin. By the way, do you guys know the single largest use of money by the federal government? No? Insulin. The single largest use of money by the federal government is our medical system funding insulin for Americans to use. Yeah. So that's actually the largest line item budget, period. Yeah. If anybody was curious, the United States actually has the best funded socialist medical system in the world. And actually spends more than any other country on a government-funded medical system. Okay? It just is not super effective. Anyways, you can look that up and talk about it in poli-sci class sometime. Very interesting topic. Off topic for this course. So, recombinant DNA also allows the insertion of genes into food plants. This affords affords crops advantages during growth and can allow us to more efficiently produce crops. There are some issues with the patenting of GMOs causing issues, and GMOs having a not very long history of actually being in use. Now, the reality is that we've been genetically modifying organisms, especially our crops, for, tens of thousands of years. We're going faster than we ever went before. And so people do get a little leery of it, and I don't really blame them. So in the late 1990s, a variety of cotton was actually introduced that has bacterial gene inserted. This produced an enzyme that was actually resistant to weed killers, and so you can plant that cotton and then spray weed killer around it to kill all the weeds. Whether that's a good thing or bad thing, I'll let you decide yourself. You can look up the relevant issues if you'd like. When farmers apply the weed killer to a field of modified cottons, you can kill the weeds, the cotton plants can survive. You can overwhelm that resistance, by the way, and you can actually poison the soil. But as long as the farmers are careful, it works pretty well. And farmers are actually quite careful and are very efficient. So never, ever doubt the intelligence of a farmer. Many of the food crops today that are in widespread use around the world are actually GMs, genetically modified foods, as the new abbreviation of it, where they have been targetedly modified since the 1970s. You probably mostly buy GMs at your grocery store unless you're spending a lot of extra money to buy quote-unquote organic, though you would be surprised if you actually look into the standards for organic, which are actually have no real definition or enforcement. So you actually might want to look into what you're buying. If you're buying organic, a little bit harder. But anyways, most of the stuff that's unlabeled is typically GMs. This is your recombinant DNA technology right here. So your basic steps to recombine DNA in order to produce a protein in another organism is going to be identifying and isolating a gene of interest. So you find the gene that codes for the enzyme or protein that you want to insert, whether that's glow-in-the-dark jellyfish or whether that's a crop that's more resistant to weed killer or a crop that doesn't get eaten as much by X bug. Okay. Then that gene of interest is located on that chromosome. It's then removed. This is going to be what's called the donor DNA. The removal is then done typically with a restriction enzyme, though there are several methodologies you can use here, but restriction enzymes usually work the best. They're the most targeted. Okay. And so you're then going to take that donor DNA and you're going to insert it into an organism DNA. This is done through what's called a vector. So a vector is going to incorporate a donor DNA into a genome of an organism by inserting itself into a new cut you make in the organism's DNA. So a vector is typically found in bacteria as a circular DNA that's called a plasmid. And so it's just a DNA that's been attached to itself to make a giant circle. Okay. Using the same restriction enzymes, we can actually open that plasmid and we can insert that gene. The vector DNA is then going to be incorporated into your bacterial DNA, and that's going to be as the bacterial divide. You're going to replicate it over and over. You'll then express the incorporated gene in the new organism, and so the bacteria is going to have that DNA that's going to be produced, the protein, on that gene that we inserted. That protein is then going to have non-native genes. That's called expression. The protein can be subsequently isolated and purified for its intended use. This is why normally we're talking about bacteria when we're doing this, because we want to produce something like insulin, right? And so an easy way to do that is to insert an E. coli, which grows very well. We can then kill the E. coli, get the insulin by extracting it. Okay? This right here is that overall process. So again, that's a DNA, donor DNA. This is the one we're going to insert in. So we go and split that. The ends of this, we use the same restriction enzyme. are going to be similar to the ends of this one. And so this will bind very easily and stick. This is then going to be the overall one with the donor. The complementary base pairs will then match up in that process of replication, just as the normal DNA would. And so then that's going to replicate that into new cells. We then have cells that will have that expression, that protein, that enzyme that can be expressed. Did everybody get that okay? Okay. Okay, I'm trying to do a summary without going like face first into the process. Okay. CRISPR. This is the 21st century gene editing. This is the newer gene editing. And what currently is sort of being further developed, they've actually gotten pretty good at CRISPR now. Surprisingly good. So scientists developed a tool that can insert DNA sequences directly into a cell's DNA. This is actually different than the previously discussed recombinant DNA because now the gene insertion can be done without the use of a vector. And the vectors are very difficult to do because these DNA fragments are not small, right? It's very difficult to get them where they need to be, especially in something like a human that has a nucleus, okay? And so instead, the DNA is going to be directly delivered to the cell that needs the new gene. This is actually the CRISPR portion of this, which stands for clusters of regularly interspaced, short, palindromic repeats. This can actually edit the base sequence of a gene without inserting DNA from another source. CRISPR was actually first discovered by scientists studying how bacteria fight off viruses, actually. And so we looked at how viruses accomplish this to develop our own methodology. Okay. When examining bacterial DNA, they actually found short, repeated base sequences identical to those of the infecting virus. Research actually determined that certain bacteria actively incorporate viral DNA into their own DNA as a defense mechanism. If a virus were to enter one of these bacteria a second time, the bacterium would use the previously incorporated repeats to produce viral RNA that matches up with the invading virus's DNA. And then those are going to pair up and inhibit the viral RNA from functioning. Kind of a cool mechanism, right? Huh. It's actually surprising what nature comes up with sometimes. Yeah. This signals an enzyme called Cas9 to bind and cut the viral DNA, rendering it useless. Scientists actually manipulated this bacterial defense mechanism in the lab to allow gene editing in cells that can be now delete, modify, replace any of the genes in the organism. And so we can kind of do whatever we want, though it can take a very long time and be very costly. And really the cost is kind of the inhibiting factor here. The amount of time and effort it takes to modify the DNA of an organism can just preclude it from actually being a real treatment. This is why, for example, the new CRISPR techniques to modify genetic diseases, they're like $6 million treatments. And so, for the most part, people can't get that. Though it is raised an interesting question over the lifetime of somebody with genetic disease. Do they have $6 million of treatment with the drugs they're taking? Yeah, probably, right? So is it better just to give the treatment up front? Huh. Anyways, moral question. You guys can answer those on your own, okay? Or take an ethics course. That would be interesting. So again, CRISPR technology is actually incredibly important, promising for changing DNA sequences, for disease-causing genes, engineering genes that perform new functions, and creating sort of like entirely new, never-before-possible combinations. There's some pretty interesting stuff that's getting used for. AI plus robotics is actually starting to make this much faster. And so it's possible this is going to get much, much faster very soon. There's already kind of hints of this. coming about. We're getting actually really good at it. This right here is actually your CRISPR technique right here, and nice pictures here. If you want to go back through and look at the pictures while you're hearing the explanations, please do. I'm running out of time, so I'm going to have to just sort of finish this one up. So this is fluorescent proteins. This is like the thing every scientist does when they're trying to play around and figure this out, because fluorescent proteins are very visually striking, and so it's easy to see that you've done it. And when they're trying to kind of learn how to use CRISPR on new organisms, this is almost always what they do. Okay. Look, I didn't mess up this organism, but now it's fluorescent. Okay. Kind of is. Anyways, this is the green fluorescent protein. Here you can see the green fluorescent switch, right? They're actually emitting light. Okay. If you put them under dark conditions, they're emitting light. And so this is sort of the test bed that's been used to do different cancer researches. We can actually edit out genes and see what it does. And thus we can find the genes that are specifically causing certain mutations. Okay, it's not just modifying the organism, it's also going the reverse. We can zap genes, stop them from functioning, and find that, oh, this is the gene that does that. That's the part of the gene that's causing the defective hemoglobin. Okay? That's why our predictions of which is which have gotten so good. This is your study guide. Let me take a vote. Who here wants to make sure I cover chapter 12 next lecture? Or who here would prefer just to call it a semester and we only go through chapter 11? Okay. Does anybody actually care if you get chapter 12? Okay. Now, I would recommend you do look at it if you're going to do anything like nursing and stuff. That's actually the chemical pathways. But you are going to get that to some degree in other courses, okay? So I'm going to leave the material. I'm going to throw it under bonus, okay? It's self-improvement. And I'll make it so you have it available, but I'm not actually going to do the post for chapter 12. I'm not going to put it on the exam. We'll call it good. Good? Okay. Anybody have any questions? Yes. Okay. Thursday, I'll be here, but we'll do a